Hi all, I'm Aparna Ratheesh and I'm an assistant professor associated with the Warwick Medical School and Center of Mechanochemical Cell Biology. And those of you who will come to us to do the MSci Integrated Science course later this year will be doing a module with me and one of my colleagues for 2 weeks, which is called Forces in Development. To be clear, this module will be about physical forces in embryonic development, dealing with the basics of embryogenesis as well as the mechanical regulation of it. But before I go into details of the module, here's a brief intro about myself. I'm an active researcher and I run a small lab. The people who you see over there is Jack, Paulina, and Diana, who work with me. As a lab, our focus is on understanding physical and chemical regulation of cell movement within developing embryos. We know now, we've now known for a long time that cell movement within developing embryos, which is a very confined space, is all about receiving signals and then further pushing and pulling and shoving their way through a very complicated environment. And to understand this process in detail, the system that we use in our lab are the macrophages. So if you look at literature, general literature, you will see that macrophages are these perpetually hungry immune cells which move around your body, eating up pathogens and dead cells and debris. And they perform very important functions both during normal physiological conditions as well as during pathogenesis, such as cancer. We study their migration in a developing fruit fly embryo, which is what you see over here. So the movie that you're seeing is a time-lapse recording of a developing fruit fly embryo. Every cell in this embryo is labeled on its membrane with a green fluorescent protein. And the purple cells that you see over there are the macrophages which are moving within the embryo. And this migration is what we study in our lab. The imaging techniques, the microscopy techniques, and the analysis techniques that we'll be using in the lab, and some of the questions that we are trying to answer within my own lab will be what we will be covering in this module. So why do we study forces in development? So Walpert, who's a very famous British developmental biologist, said that it's not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. Gastrulation is one of the most important events of embryogenesis. I'm not sure I completely agree with the sentiment, but I definitely believe that it's not just birth, marriage, or death, but embryogenesis, which is the most important time of your life. We know this is true if you look at the number of infants that are born across the world with birth defects, which happen because of processes that are going wrong during development. And so clearly understanding development is very important in tackling these issues. But there's another reason why studying development is very fascinating. It is because now we know that a number of pathways which are actually going wrong during pathogenic conditions, such as inflammation or cancer, these signaling pathways have actually originally occurred in the embryo during development. So a lot of what goes wrong in pathogenesis is recapitulation of processes from embryonic development. So understanding development also gives you a handle on understanding these pathogenic processes better. So now, again, to go back to embryogenesis, what is embryogenesis? This is the entire process where you go from being a single cell to an entire being. So clearly it's a very incredibly complex process which involves a lot of genes, proteins, and signals. And this has been studied for a very long time. But now let me show you a movie of a developing fruit fly embryo. So each spot that you see is one nucleus. And you're seeing two views from here, from the top and from the bottom. And when you watch this for a while, what becomes really obvious is that along with the biochemical signaling that's probably going on, it's also a very mechanical process. You have to imagine that this is a lot of cells cramped into a very small space, and they have to do a lot of maneuvering in that little space to get to the right place, to set up tissues, to set up populations, to set up organs, which is ultimately what happens during embryogenesis. When I watch movies like this, it brings to mind images like this, which is also all about maneuvering in cramped spaces. And of course, since I was born and brought up in India, it brings to my mind images like this, being stuck in Indian railways. So while it's clearly obvious, both from the embryogenesis movies and from these images that I'm showing you, that it's not just about signals. Physical forces must play a huge role in the way cells move and position themselves in the embryo. The same way every push and shove kind of brings you closer to this elusive door when you are stuck somewhere when you're traveling within Indian railways. So, as I said earlier, along with gene expression and signaling, in the last decade or so, physical forces have been identified to be one of the most important regulators of development. And these are not physical forces that are hard to understand, these are very intuitive and obvious. As you can imagine, the cells feel forces such as tension, like what you would feel if you were to stretch a rubber band. They would feel compression, they would feel shear, and the cells feel these at a single cell level and at the level of tissues. And the propagation of these forces over long range tissue ranges is very essential for development. So, in order to understand the role of physical forces in development, in this module, at least to understand a part of it, we will be having both lectures and lab sessions. And we will take an interdisciplinary approach to this problem, which, to be honest, has evolved very organically in the field of cell and developmental biology. If you look at an average cell and developmental biology lab these days, you will find at least one physicist, one mathematician. This is just the way the field has evolved, and the tools that you need to understand these processes in detail has actually come from multiple disciplines. So in the lab sessions, we will take these embryos and we will exert different kind of forces on these embryos and try to understand the consequences of these using microscopy and image analysis. So again, like I said, we will be wading through a number of disciplines to get these answers. For example, you will start with taking the live embryos, exerting forces on these, like compressive forces, like sucking them up into pipettes. And in the lecture sessions, you'll be understanding basics of this process from the cell biological, biochemistry, and biophysics aspects of it. Later on, you will go on to do microscopy of these treated embryos, to speak. And in the process, you're going to learn more about optics and chemistry. And finally, finish up with image analysis, which is all about mathematics and programming. I think it's something that's very clear to all of us when we watch news these days, especially about COVID-19, is that science has become very interdisciplinary in the real world. So if you ask people earlier, what do you think, who will be tackling pandemics and vaccine development? It would have been thought to be the realm of biologists. Now we know that's across the world where lab biologists are working with modeling experts, with engineers, with chemists and physicists to come up with real world answers, to come up with answers to these real world problems. So clearly, interdisciplinary science is the way forward to tackling problems in our world. So to sum up with, this module will prepare you to gain some knowledge across these areas. So basic understanding of embryonic development, the forces involved in it, the forces that are common during development, and we'll go into details of these. We learn about handling live samples and imaging on a tissue and an embryonic level, followed up with image analysis and computing, and you will end this module with some scientific writing, where you write up your findings in the form of a manuscript. So you will be learning a number of tools which you can use across multiple modules, and ideally later in your life across multiple disciplines. And that's all I have for you today. And if I can leave you with a very popular manuscript title in developmental biology, may the force be with you. Thank you.